Well, good evening, everybody, and a warm welcome to this evening's BBSRC Anniversary Award Lecture for Excellence in Bioscience. Before I introduce tonight's speaker, Professor Caroline Dean, I'd like to say a few words about the lecture and the award itself. So the Biotechnology and Biological Sciences Research Council, or BBSRC, is the principal funder of non-medical life sciences research in the UK. And to celebrate the 20th anniversary of its formation, last year it instituted three Excellence in Biosciences awards, which were to be given to outstanding scientists operating in any area across the field that BBSRC sponsors, that is, any area of bioscience which is not medical sciences. So the conditions for the award were, first of all, you had to have done outstanding research, but not only that, it had to be research with great originality and with impact right across science, and it also had to have some impact on society or policy. And as I said, there were three awardees. Each awardee was given the chance to give a public lecture and awarded £2,000. And tonight is the public lecture, which will be given by Professor Caroline Dean from the John Innes Centre. So let me say a few words about Caroline. She started her career at the University of York, where she did her bachelor's degree and her PhD. And after that, she went and worked in California for 5 years in the private sector in the newly emerging area, the then newly emerging area of plant molecular biology. Having been really one of the pioneers in this field, she arrived at the John Innes Centre in 1988, where she has been ever since. And I think her work through that period has been, through her period at JIC, has focused really with beautiful insight into the way in which plants know when to flower. How does a plant know when it should start flowering? And that's not a trivial problem. And as I hope you'll hear tonight, that's been a journey for Caroline, which has resulted in numerous discoveries of the way that plants have memory and the way in which they signal and sense the environment and deliver the desired physiological result. So Caroline has been the recipient. I wanted to just to say also that some of the impact. I wanted to point out two aspects in which Caroline's work had unusually broad impact. One of which was that during the 1990s she developed a lot of tools, molecular tools, for the wider plant community using the model plant Arabidopsis thaliana, which you'll hear more about tonight, I'm sure. And Caroline's influence then spread way beyond the simple question of how does a plant know when to flower. The tools that she gave to the community were widely used and very valued, and helped us in all sorts of areas of plant biology way beyond flowering. And then the second aspect in which she's had wider impact is that some of the mechanisms that she's discovered in plants have actually laid a basis for understanding epigenetics in all life systems. And I think that is a really fascinating aspect. Often it's seen that plants somewhat lag behind animals or human research in the way that we develop an understanding. But here is one of the famous example of plants leading the way. So Caroline has been a recipient of numerous awards. She was awarded the Genetical Society Medal in 2007. She's been elected to the Royal Society, as well as a foreign member of the National Academies of the USA and of Germany. She's an elected member of the European Molecular Biology Organization, and just this year EMBO, the European Molecular Biology Organization, and FEBS, the Federation of European Biochemical Societies, awarded Caroline their prize, which is awarded to a single female scientist across the whole of Europe who has not only conducted excellent science, but also proven to be a wonderful mentor for the new generation of scientists. So with that, I'll introduce Caroline to give her talk on how do plants remember. Caroline.
Well, thank you, Dale. Thank you, everyone, for coming for the public lecture. It's great timing to do a public lecture like this because what I'm going to talk about is spring flowering. That's the process that my lab has been studying. Now, plants flower in spring because conditions are better for pollination, for seed set. But you might ask yourself, how does a plant know when it's spring? Well, if you think about it for a minute, you think, well, it's going to warm up a bit. Maybe that's how it knows it's spring. Cold's over, now it's warming up. And that's part of the answer, but it's not enough because actually, if it relied just on that, very occasionally in autumn, it would get confused. A nice week in November would horribly confuse the plants, and flowering then would be disastrous because they would be flowering in the middle of winter. So plants use many different environmental signals to actually figure out when to flower. And they monitor those signals and they remember them, hence the title, How Do Plants Remember? Now what I'm going to do today is give you an overview of the kind of research that's going on in the lab, looking at this general question of how do plants know when to flower. I'm going to try and explain why it's so precisely controlled, and it is very precise. If you look at a row of cherry trees, and I meant to say in the previous slide, that just shows how precisely the flowering is controlled. All the trees in that row of cherry trees will probably flower within a couple of days of each other. It'll be different from a different species, but each tree has taken all of the information from the environment and integrated it and made the decision to flower almost synchronously. So I want to tell you about how they do that. I want to tell you a little bit about what they monitor and how that is remembered and the basis of memory in plants. And then lastly, I'll turn to adaptation. So if plants have understood what the different environments are, how does that change then when plants adapt to different environments? So hopefully by the end of that you'll have a bit of a clue about what we're doing and hopefully I won't confuse you too much. So how did I get to this topic? You know, it's always interesting to look back and think, well, how did I end up studying flowering time research for 25 years now? Well, I was very into doing experiments when I was an undergraduate. Loved doing lab practicals. So I found a picture of myself looking at doing some plant biochemistry. You can see a younger me, aged 21, mouth pipetting, which of course would be absolutely banned now, some extract that I'd made from some plants. And because I loved doing lab work, I then stayed and did a PhD in the same lab. And during the time I was doing a PhD, my supervisor went off to California for a sabbatical visit. And she came back absolutely raving about it. The science, the culture, the sun, everything. And I thought, wow, you know, here was I, a PhD student in York. Very nice place, York. Dale was at York as well. But, you know, California sounded very intriguing. So I applied for some positions and got a position and then took myself off and had 5 years in California. As Dale said, it was just at the beginning of this whole era of genetic engineering. And so it was with a small biotech company that I started working, Advanced Genetic Sciences, in Oakland, California. I did have a really fantastic time, but one thing that struck me very quickly when I was there was just how different seasons were in California. Never really thought about it, you know, having grown up in Manchester as a schoolgirl and then York as a PhD student. The seasons there were very clear, very striking, and the plants followed the seasons, and the development was aligned with those seasonal changes. It was very different in California, and the plants then behaved differently as well. So my interest in this whole seasonal information and how it affects plant development was cemented when I went and bought some tulips at the nursery garden in California. I was feeling a bit homesick. I thought it would be nice to have some tulips on the balcony. And the person in the shop said to me, now don't forget to put them in the fridge for 6 weeks. And that really stunned me. You know, not only were they monitoring these seasonal cues, they actually needed to have them to actually have the developmental processes working. And so when I had the opportunity to run my own lab, I thought, yes, I'll tackle what is in the seasonal information that allows plants to flower when they do. So I came to John Innes Centre in 1988 and said, yes, I want to study vernalization, the acceleration of flowering by prolonged cold. Now, we knew quite a lot about vernalization because it's so important for crops. There'd been a lot of research in a lot of our brassica crops and in cereal crops at the John Innes Centre and the Plant Breeding Institute, which had moved very recently from Cambridge to Norwich and formed part of the John Innes Centre. A lot of genetics in developing winter and spring varieties of these major crops basically bred vernalization. If the crop is sown in autumn, it has to overwinter vegetatively and then flower in the spring. It's a winter type. It needs this prolonged cold or vernalization to be able to flower. Alternatively, there are spring varieties which are sown in March, April and don't need vernalization. So winter wheat, spring wheat is probably familiar to many of you. So there's a lot of information about vernalization at the physiological and genetic level, but absolutely nothing at the molecular level. It was also extremely difficult at that point to figure out how we were going to tackle the molecular understanding in crops. So at that point, I joined a sort of an emerging sort of front in plant biology where there was this decision, rather than studying lots of different plant species, where it was quite hard to compare different results from one lab in one plant, from a different lab in a different species. There was a move to all concentrate to focus on one plant species, a plant that would tell us a lot about all plants. But by concentrating on one, we could then compare everything, and all the tools developed in one lab would then be usable and useful for everybody else. A lot of discussion about what that plant was going to be, and as Dale said, it turned out we chose Arabidopsis thaliana. Now, the common name for Arabidopsis is thale cress, and I can guarantee it will be in your garden. It's just about to flower, probably as it warms up next week. It'll all start flowering. It's quite small, as you can see from the 50p size. That's what you should be looking for in your driveways, in your side of the garden. It's looking really good this year. I carefully weed around it in my garden, sort of prized Arabidopsis, watching what they do. It would be great to have a big collection. We can do a natural variation analysis of Norwich Arabidopsis thaliana. So please collect them this year, send them in. So now there are probably, I don't know how many labs study plant biology internationally, but probably 80% of those labs in the world all work on this one plant species. It's extraordinary the progress that has been made because of that concentration of effort. Now, why was it chosen? It's small, it's easy to grow, but it's also got a very small genome. Now, you'll have heard a lot about the human genome, the DNA in each of our cells, and how that's all been sequenced and how that's going to affect, our diagnosis of disease and things. The genome in Arabidopsis is tiny. It's about 50-fold less than a human genome in each cell, the amount of DNA. But it's got about the same number of genes. And you might be surprised to know that about 50% of those are really the same in plants and in animals. You can take a gene out of a plant and put it into the human, and it will do exactly the same thing. So down at the DNA level, plants and humans are really rather similar. But we could use all of those. We developed tools, and as part of that, we could study this flowering process. It responded to all of the normal environmental cues plants respond to, light, photoperiod or day length, and this requirement for prolonged cold or vernalization. But because of this very small amount of DNA in each cell, we could then clone the genes. And that's a technique, molecular genetics. That's the first time it was applied to plants was in Arabidopsis. All you need to know about those genes is what happens when they go wrong. And based on that information and the molecular tools, you can clone the gene out and ask, what does that gene encode? What kind of protein does it make? And therefore, what is the molecular basis of that function? So that's what we did. We started off There were several groups working on flowering time. We looked at mutants, so we made plants now that flower differently by doing chemical mutagenesis, where we disrupted specific genes, and we characterized a whole series of genes that were required to switch the plant to flowering. So the plant on the left is an Arabidopsis, it's not flowering, and the one, and I show a schematic here of a flower. I'm just going to give you a schematic. So there's a whole series of those, probably 500 or so in the genome, that actually are required for the little growing tip of the plant to switch from making leaves to making flowers. Now, their regulation is absolutely key to the flowering process. And its control, they are controlled, or they're activated by these various environmental cues. Photoperiod, day length, light quality, temperature, all switch those genes on. So the arrows mean activation in this case for these genes. But that's not the only story. There are a whole series of genes as well that keep flowering off, keep those genes switched off. So there's this continual push me, pull you going on to decide whether or not the genes that now switch the plant to flowering are switched on or not. You're going to hear a lot about this flora repressor. I'm going to refer to it as a brake. This brake keeps the genes off. And what winter does is to switch off that brake. Now, this is a relatively simple schematic, and I just want to walk you through it. So an analogy that I find quite useful looking at this is as if you're in a car. The car wants to move forward, and that's like flowering. You have various pedals. Some of those pedals accelerate the flowering. Some of those pedals prevent the flowering. They're brakes, prevent the car moving forward. And so it's the balance of activities that actually you have to take your foot off the brake and press the accelerator before the thing can move or the plant can flower. So in fact, all of these pathways are working all at the same time, and it's the balance of their activities that then determines whether the plant switches to flowering. So because of my tulip moment, of course, we focused on the pathways involved in winter, how winter switches off this brake, which in turn helps the plant to flower when all of these other promotive pathways are present. That's what vernalization is. Now, if a plant needs vernalization, it's going back to what I was saying about winter and spring sown wheat, they have what's called this winter annual habit. So if you need winter, you have to germinate in autumn, over winter, and flower in spring. And an alternative strategy is not to need winter, and then you basically germinate in spring and flower that summer. Now, if you look at a lot of plants, and I'm sure many of you are gardeners, different plants have adopted different strategies. This decision is very important for adaptation of both native plants and also many crops. And in fact, for a lot of crops, the development of winter and spring varieties has really significantly extended geographical range of production of those crops. Now, in Arabidopsis, we have similar winter and spring varieties. The one on the left just keeps producing leaves. This is grown in our nice warm growth room. It wouldn't look like this out in the wild. But if you put it in a nice warm growth room, it thinks everything's fine. It just keeps making leaves. It's not had the information or the signals to say I've had winter, so it just keeps making leaves. And they get bigger and bigger and bigger. However, if you put seed of that type in the cold for 6 weeks before you put them in your growth room, then they look like the plant on the right. So they have a requirement for this prolonged cold to then accelerate the switch to flowering. So some need cold, others don't, the summer annual types. Now, when we looked at this was the first question we looked at because, of course, it was so important for many of the crops, this winter-spring difference. What we found was that it was actually quite simple when you got down to understanding the mechanism. And this gene, this brake gene, this repressor I was telling you about, its name is flowering locus C. Not a very easy name to remember, not a name I gave it, but I have to live with it. It's become the center of my universe, as lots of people who know me will tell you. If you express this gene and make this protein, this brake protein, it keeps all the genes off that are required to switch the plant to flowering. It's the brake in the car. And I do see FLC everywhere. I even saw it last week opposite Strangers Museum. There's up near St Andrews, so I had to take a picture. And actually, someone in my lab found a license plate, FLC1, and they say, rushing in, said, Caroline, really, this has to be your license plate. But I thought that was a step too far. Now, remember FLC. We'll keep coming back to FLC all the way through. FLC is the center of the repression. Now, in the winter annuals, FLC is really actively switched on by a second gene called frigida. Again, this was named in the 1950s by a German professor because it's the gene that means the plants need cold. And so my analogy in the car is frigida is the foot pushing hard on the brake. If you have frigida, you push hard on the brake, that plant is not going to flower. And what was very interesting, when we looked in the summer annuals, so these are the ones that need winter, the summer annuals all had a broken frigida. In fact, when we looked in different summer annual types, frigida had been broken in different ways. So evolution had got rid of this leg every once in a while when it was useful and adaptively significant to actually be able to switch from having to go through winter to being able to flower much more rapidly. That was a very big surprise when we first discovered that. What cold does is reverse the effect of frigida. It doesn't affect frigida itself. Cold directly switches off the brake, FLC. So really, this very complicated physiological response, when you get down to it at the gene level, can be seen as relatively simple. So now I'm going to turn to actually vernalization and the process of how plants perceive the cold and remember the cold, hence the title, How Do Plants Remember? As I said, what cold does is switch off the brake itself. Now this is the characteristics of vernalization and Arabidopsis, but I could have been putting any plant up here, wheat or seed rape. They have a very similar set of characteristics in that it's a quantitative process. The more cold the plant experiences, the faster the acceleration. So I hope you can see that these plants have been given 3 weeks or 6 weeks of cold before they're put in our nice warm chamber. And you can see that there's a graded response in flowering time. The ones on the left will just keep making leaves, they've had no cold. The ones on the right have flowered quite quickly, and the ones in the middle are the sort of Goldilocks, just right, halfway in between. So it's a quantitative process, and that's quite interesting. And actually, it's quantitative, and it's important it's quantitative because it needs the whole of winter and not just a couple of days of cold. For the reasons I said before, it needs to be able to make sure that the flowering doesn't occur until the favorable conditions of spring. The second and really important characteristic is that it's stable through cuttings. So if you take a plant, give it the cold, and then take a very small part of that plant and regenerate a new plant from it, it will remember it's experienced the cold. Something in those few cells that you've used to make that cutting holds the memory of winter until the whole plant regenerates and it flowers at the appropriately early time. That really intrigued people. What was this memory? How did the plants feel this period of cold and remember it? But it's always reset through seed, because obviously every generation of plants wants to make sure, if it's a winter annual, that every generation is going to need to have vernalization. So this was made easier to understand when we actually looked at the expression of the brake gene. So this is our gene FLC, remember? This is the brake, keeps all of the genes off required to make a flower. When we look at it, and you're just looking at black here, this just happens to be the amount of activity from that gene. So there's a lot of it in plants with 0 weeks of cold having been given. And as plants experience more and more cold, the gene progressively switches off. Now seeing that explained why this whole process was quantitative, because the cold was slowly switching off this repressor and slowly then taking the foot off the brake. It wasn't an all or nothing thing. But the other thing that was interesting was if you took the plants out, let's say after 2 weeks cold, and then grew them on, the level of FLC stayed at whatever level it had gotten to by the end of the cold. So if you took it out at 1 week, it stayed at 1 week's level for the rest of development. 2 weeks cold, it stayed at 2 weeks level. And that memory of the quantitativeness of the silencing gave the characteristic that this is what's called an epigenetic process. Now, I decided I was going to try and really explain epigenetics because it's huge. Now well, hopefully, after I've explained it to you, when you go to the newspapers, you'll see it everywhere at the moment. It's important for most development of most organisms, and therefore very important for disease of many organisms, including humans. So epigenetic is the layer above genetics. Now, I've pinched a schematic from the internet here where I've unraveled a chromosome. So the chromosome at the bottom, remember the chromosomes are the various pieces of the genome. In humans, there are 23 chromosomes in each cell. In Arabidopsis, there's just 5. So if you take a chromosome and unravel it, what you see is that it's made up of the DNA double helix wrapped around protein beads. These beads, they look a bit like a necklace. The DNA wraps around them. And either the protein beads or the DNA itself can be chemically modified. There's a whole range of different modifications. What's shown at the top is methylation of the DNA. The bases themselves have got a methyl group added to them. So either modification of the base or the protein has a huge effect on the expression or the activity of that gene. And people have compared this to hardware and software. So if you understand computers, this may be useful. If not, bear with me for a minute. The DNA in each cell, of course, is the same. The DNA in every cell in our body is the same, in all the plants is the same. That's the hardware. You need that. That's the set of instructions. But of course, every cell doesn't always do the same thing. It needs instructions to use the hardware in a specific way. And that's what software does. It instructs the hardware to make a program to draw a graph in the computer or to write a document. In the plant's case, the software changes the DNA to say, switch these genes on, and then keep them switched on until I tell you to switch them off. So the modifications of the DNA and the proteins are absolutely what is driving the development of bodies, of plants, and any time they're broken is when we get disease. You'll see it a lot, as I said, in the mainstream press. These are just a couple of examples where people got very excited because, and many of you may have read this, there were some reports where what we eat, and in fact, actually during World War II, when there was famine and they monitored pregnant women, they found that the famine was affecting the fetus, and in fact, the children of those fetus were also affected. So somehow there was a memory through generations of what had happened and how much nutrition the mother had had. And so you could imagine this is true for many things we take on board, affecting our DNA, and those are heritable changes. If they go through to the fetus, then they're inherited to the next generation. So this is what the press got very excited about. But epigenetics really is not just transgenerational effects, it's what drives the development of our bodies. It's what keeps all of the cells doing the right things. In the embryo, the cells are given instructions, make a hair cell, make a bone cell, and all the daughters of those cells stay as hair cells or bone cells without any additional instructions, because it's the changes in the DNA and these protein beads that are stable and then replicated through the different generations. This is the epigenetic regulation. A lot of studies of identical twins tell us what is controlled by epigenetics, because of course they have identical genomes, so any differences have to be epigenetic. And the epigenetic regulation is often the basis of many cancers. The BRCA1 gene, very famous gene, is actually required for a healthy formation of various DNA repair mechanisms, in fact. And when you lose that and it's lost through changed epigenetic regulation, there's a very high incidence of breast and ovarian cancer. Same is true for colorectal cancers. And it's having an enormous impact on cancer diagnosis and therapeutics, the understanding of this epigenetic regulation. Okay, so let's go back to our plants. How is winter remembered by this same kind of mechanism that's so important in our bodies? So what I've talked about so far is actually changed methylation of the DNA itself. In fact, what we found for the memory of winter is it's the necklace, it's the bead modification that's important for memory of winter. And I'm going to show a schematic where this, my PowerPoint skills were not up to drawing one of these beautiful red things, so forgive me. Instead of now a red bead, we've now got a yellow bead, but it's the same idea. It's one of these small pieces of the chromosome taken out. That is what's called a nucleosome, the DNA wrapped around a protein bead. Under a microscope, it actually looks a bit like a necklace. You can see on the left the protein beads with the DNA wrapped around. And these beads have modifications. They're either the various different modifications, and I'm just color-coding them with a traffic light system. So a green modification says to the cell, this gene should be on. So all the proteins come that are required to switch that gene on. A red modification, in this case, traffic light stop, means to the cell, this gene should be off. And so the different genes are covered with these different colored modifications. And what happens in flowering now with our break gene, FLC, we have our green modifications in autumn, the gene is on, the plant won't flower. Plant begins to feel the cold, the gene is switched off, and the modification is now switched to red. That says to the cell, keep this gene off. Now winter's gone past and it's spring. Okay, so what are the modifications on our FLC gene now? So this is a kind of little test halfway through to see if anybody's understanding. So we'll have a vote. It's either green or red. Who thinks it's going to be green? Few. Who thinks it's going to be red? Yay. It's red. I haven't confused everybody. What happens is that there's this memory, these epigenetic modifications are heritable. They go from daughter to daughter to daughter without the initial signal. The gene is switched off in the winter, but then it stays off. Even when the plants come into the warm, it stays off, allowing the plants to flower. Now, as I said before, it requires a prolonged period of cold. It needs long cold. And it needs long cold because otherwise it's going to make a mistake and flower too quickly in autumn. And actually the mechanism, the way it does that is really interesting in that I'd shown you the picture at the top where you saw this slow, progressive switching off of the break gene. And when we first looked at that, we thought, well, it's just slowly switching off slowly in each cell. Sort of what the intuitive interpretation was. But when we dug into the mechanism a bit more, it turns out that wasn't right. What was happening was each cell separately was feeling the cold and deciding, have I had enough cold? And at relatively low probability, when it felt it had enough cold, it would switch the gene from the green state to the red state of FLC. And that switch happens slowly, 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 so you can see that with increasing weeks of cold, the number of cells that have made the switch is now increased. So this is my schematic of a plant. Blue means the gene is on, white it's off. But if you grind that up and then look as it above, it looks like a gradual change. But it's not gradual in each cell, it's the number of cells that are switched that changes. So we then had a reasonably good idea that plants were feeling the cold, there was a decision to switch, and then we understood, once it had switched, how that was maintained for the rest of development. So we were sort of progressing in our understanding. But so far I've sort of talked as if winter's one thing. You know, winter being, it's 4 weeks in a cold room for most of these Arabidopsis plants. But of course winter's very different depending on where the plants are growing. And an Arabidopsis thaliana has a very wide geographical range. You can collect Arabidopsis from up in the north, at the Arctic Circle in northern Sweden, all the way down to the Cape Verde Islands near the equator. And there's been an enormous change in their flowering behavior to allow them to adapt to such different climates. So you see some of them collected from the various places. The ones from the northern latitude tend to be winter annual types. They tend to overwinter. Flower, set seed before the drought of the summer. The ones in the south actually tend to be summer annual types because actually they can get more than one generation a year. So that's really very good if you're a plant to generate twice as much seed. So there's an enormous amount of natural variation for winter, summer annual types. But what about for the length of cold? No one had looked at that before. So we made the most of the fact that they're in such nice places and decided we ought to go to the very furthest north the Arabidopsis grows to find the most extreme winter, to see just what had changed to allow adaptation to that very extreme winter. And that extreme winter was in a place called Norwich in northern Sweden, 62.5 degrees north. This looks absolutely beautiful. I've been there twice, and every time it's absolutely glorious. And I think it's always like this, but clearly not. Very nice part of the world. We've done a lot of collection there. And then we took the Arabidopsis from there and brought them back and actually put them in the growth room and compared them with, in this particular case, an Arabidopsis from Edinburgh. So on this map, it's a world map here, all the orange dots are places that we've got Arabidopsis from. So here we've compared the Arabidopsis from this northern Swedish Lervik isolate and one from Edinburgh. And what you can see is that actually the Edinburgh one, 4 weeks was enough to cause flowering, whereas in northern Sweden, that accession needed 12 weeks. Now, when we went in and looked at the behavior of our break gene, so I've drawn it here, this is the amount of FLC expression, high in autumn, then it starts to feel the cold and the gene begins to switch off. And remember, it stays off. Once it's off, it stays off through spring and summer, allowing flowering to occur. That's what happens with the Edinburgh type. But if we put the northern Swedish type in the same conditions, actually it gets it wrong, and it switches the gene back on. So 4 weeks was not enough to trigger the epigenetic memory in this northern Swedish type, because that northern Swedish type has adapted to requiring much longer winters, because that's what it's had to get used to. However, if you give the northern Swedish type 12 weeks, that is enough. So all of the proteins are there, the machinery is there, it's just adapted to needing longer cold. And when I go back to the schematic I showed before, we now know that it's that very first step that is affected, and there are just 4 simple changes in that break gene that distinguish whether you need 12 weeks or 4 weeks cold. So again, when we get down to understanding it, actually the differences are really rather small, and it's all relatively simple to explain. We still don't really know, and we're still working on this, how those 4 simple changes actually make the plant need 12 weeks. But it's an important question to actually really understand, how are the plants sensing the cold? And so this has evolved into a much bigger project now, looking at the whole of Sweden, and we've gone back. This is a Google map, and all of these green dots now are where we found Arabidopsis in Sweden. And there's very impressive natural variation within the Swedish Arabidopsis accessions, and we're working our way through trying to understand what has changed at the break gene, and it is at the break gene only, that's allowed all these different types just to slightly tweak their flowering to best fit the environment that they're adapted to. Now, part of that, and we've had a recent grant, is actually to send back plant material every month from Sweden, so we can monitor the break gene sort of through real-time winter in northern Sweden. And unfortunately, Christina is an MSc student who's based in Sweden, was actually arriving today, but she didn't get here in time to come to the talk. But this is her, and this is Lervik, the same place I showed you in that beautiful sunshine, but now this was about a month ago. And she's had to dig, 3 feet of snow to actually be able to harvest these plants for us and send them back. The plants are absolutely fine. They're quite healthy under all of this snow. And so we're doing this real-time experiment to try and understand now, what is it? What are the temperature antennae that actually can perceive the different cold? And on the left is with some temperature loggers actually showing the different temperature. And it really does vary enormously during the whole year. You can see you might think, well, okay, from November to March, that's obviously winter. There's been a period where it's all under the snow, which is why actually the temperature is much more constant. Some of the snow blew away in January, which is why it got even colder. But one surprise was that actually these plants in northern Sweden, it's too cold between November and December. They just go into a suspended animation, nothing changes. And actually they've had to adapt their vernalization so that it's much, much earlier, actually in autumn. And then they just sit and wait until things warm up again and the day length gets longer, and then they flower immediately, the snow goes. So subtle changes just make all the difference to adaptation. Now it's really important to know how plants are perceiving and how big the changes are for these adaptive changes, how they're perceiving the cold. Because of course what we're having at the moment is a warming climate, but not necessarily any changing photoperiod or day length. And so plants that have naturally evolved to monitor both day length and temperature are getting confused because those signals are changing separately. So that's causing a very significant disruption to lots of ecosystems, so lots of our crops. And I show one example of actually, do you remember in 2010 we had that really, really cold winter where there was a satellite where the whole of England was covered in white. So over that period of July to December of that year, the number, so this is from WeatherQuest, they actually, for farmers, to tell people what the plants might be feeling. So the number of chilling hours, so the number of hours below 5 degrees. So you can see on the left that they've had a lot more chilling hours from the heat map in 2010, 6 months. On the right shows basically the same time period, but just a year later. It was really a lot warmer. And so the amount of chilling those plants got was significantly less. So we're trying to develop varieties of crops that we can harvest in a sort of normal schedule for delivery to the supermarkets. And yet the temperature is changing all the time, and therefore when these things bolt and flower, it keeps changing. And we had a pretty disastrous year, you can see from the BBC News website here. All the cauliflowers in that really cold year bolted all at once. And then there was nothing for several months afterwards. So ideally what we want to do is actually develop plants that are slightly less sensitive to these temperature fluctuations so that we can actually maintain the production of all of these different crops. Okay, I've whizzed through that. I hope I haven't confused you too much, but there are some take-home messages. So if nothing else, these are the sort of 3 sort of summary points I'd like you to take home. One is that plant biology really, had a step change in acceleration and understanding when the community adopted the use of a reference plant, this reference plant Arabidopsis thaliana. And please do have a look at it. It's an extraordinary plant. When people now have gone and looked at other plant species, traits like flowering, disease resistance, height, you name it, they're all controlled in the same way in all plants. They're just variations on a theme. And by understanding it in one plant, you very much more quickly understand it in all plants. So the second point is that this enormous physiological complexity underlying flowering time, and in the garden, lots of things, they all flower at different times. Some need short days, some need long days, some need temperature. You would have thought it would have been a very complicated molecular mechanism that was different in each type. Well, that's not right. It's actually the same mechanism, the same hierarchy of genes. It's just the inputs are slightly different, taken different predominance. So evolution has just tweaked things rather than change things. And the third thing is that actually plants, yes, do remember, and they can remember by this epigenetic mechanism where there are changes to, in the case of winter, the beads, the protein beads, the DNA wraps around. And that epigenetic mechanism is really important in a lot of organisms, including humans. We still don't really know so much about it. So the plant system is a great tool to actually begin to understand epigenetic regulation, which is generally important in all organisms. And that just leaves me to thank the funders, the BBSRC, for the prize. I did say to my lab we might have a party with the £2,000, and they seemed to think that was a good idea. The other funder is the European Research Council, and very generous, helps fund the Swedish experiments. And this is my group, along with Martin Howard's group, and we had a retreat in Go Ape last summer. So this is me being made to go through all of the trees, and I don't know if any of you have done Go Ape. But we had a morning doing that where we all got together, and then we had a retreat where we all then discussed science for the rest of the day. And it's an amazingly talented group of people from all over the world. We have people come on postdoctoral fellowships, starting from the right, Portugal, England, China, Australia, Argentina, Chinese, and Israeli at the end. So it's a great group of people, and it's a great career, a really nice way of spending one's life. So thank you very much for coming, and thank you for your attention.